Welcome students in Alka's Botany channel. Please like, share and subscribe the channel. Today we are going to learn mitochondria, an important cell organelle in plants. I am Dr. Alka Inamdr in Alka's Botany channel. Mitochondria were first described by Richard Altman in 1890 and called them as a biomass. Later on, Carl Benda in 1897 coined the term mitochondrion. The term mitochondrion is derived from a Greek word mitos means thread and chondrion means granule-like structure. Its size ranges from 0.5 to 1 micrometer in diameter. Mitochondria are oxygen-consuming ribbon-shaped cellular organelles of immense importance floating free throughout the cell. Mitochondria are motile organelles that have a double membrane structure. It has 4 distinct domains: the outer membrane, the inner membrane, the intermembrane space and the matrix. Typically, there are about 2000 mitochondria per cell, representing around 25% of the cell volume. This is the ultrastructure of mitochondrion, in which we can easily see the outer membrane, inner membrane. In between these two membranes there is an intermembrane space, and inner membrane shows foldings. These foldings are at the inner side, that is why it is also called infolds, and typically these are called Cristae. Then on Cristae there are F1 particles or F1 units. Mitochondria has its own DNA and ribosomes. The mitochondrion is a double membrane rod-shaped structure found in both plant and animal cell. The structure comprises an outer smooth membrane, an inner folded or tubular membrane which has a large surface and encloses the matrix space, and a gel-like material called the matrix. The outer membrane and the inner membrane are made up of proteins and phospholipid layers separated by intermembrane space. The outer membrane covers the surface of the mitochondrion and has a large number of special proteins known as porins. Porins in the outer membrane allow small molecules to be exchanged between the cytoplasm and the intermembrane space. It is freely permeable to ions, nutrient molecules, energy molecules like the adenosine diphosphate, that is ADP, and adenosine triphosphate, that is ATP molecules. The inner membrane also contains proton pumps and many permease proteins for the transport of various molecules such as citrates, ADP phosphate and ATP. The inner mitochondrial membrane is rich in many enzymes, co-enzymes and other components of electron transport chain. The inner mitochondrial membrane gives out finger-like infoldings called Cristae. Singular, it is called Crista, which contains tennis racket shaped F1 particles that contain ATPase enzyme for ATP synthesis. We can easily understood by these diagrams, whereas mitochondria, outer and inner membrane, and on inner membrane there are F1 particles contain ATPase enzyme. The inner membrane is completely impermeable even to small molecules except oxygen, carbon dioxide and water. As a consequence of the greatly enlarged surface area, the inner membrane can contain more than 50% of the total mitochondrial protein. Mitochondrial matrix is a viscous fluid that contains a mixture of enzymes and proteins. It also comprises ribosomes, inorganic ions, mitochondrial DNA, nucleotide cofactors and organic molecules. The enzymes present in the matrix play an important role in the Krebs cycle electron transport system and synthesis of ATP molecules. In this diagram, this blue colored gel is nothing but a mitochondrial matrix. Hydrogen ions reduce the molecules of NAD and FAD, both of which pass on hydrogen ions to respiratory or electron transport chain, where oxidative phosphorylation takes place to generate energy-rich ATP molecules. Since mitochondria can synthesize 10% of their proteins in their own protein synthetic machinery, they are considered as semi-autonomous organelles. And all these are functions of mitochondria. The most important function of mitochondria is to produce energy, and so popularly known as powerhouses of the cell. Mitochondria produce energy, store energy, and also supply energy to the cell for various cellular reactions and mechanism of the cell. That's why it is called powerhouses of cell. Mitochondria produce the molecules adenosine triphosphate, one of the cell's energy currencies that provide the energy to drive cellular reactions and mechanisms. They play a major role in breaking down nutrients and generating energy-rich molecules for the cell. The simple molecules of nutrition processed in mitochondria and produce charged molecules. These charged molecules combine with oxygen and produce ATP molecules. This process of formation of energy-rich phosphate bonds ATP with the help of oxygen, that's why it is called oxidative phosphorylation. Mitochondria help the cells to maintain the proper concentration of calcium ions within the compartment of a cell. The mitochondria also play an important role in process of apoptosis or programmed cell death, whereas during apoptosis, cytochrome F releases from mitochondria and it breaks down the supply of energy for that apoptotic cell, and due to that programmed cell death occurs. It is also involved in various cellular activities like cellular differentiation, cell signaling, cell senescence, controlling the cell cycle, and also in cell growth. Mitochondria is considered as a semi-autonomous body because mitochondria contain circular and slightly twisted DNA as well as ribosome and polyribosomes in their matrix. Maximum portion of this DNA is used for protein synthesis. It synthesizes rRNA, mRNA and tRNA of about 10 to 12 mitochondrial proteins. The growth in mitochondrion is the integrated activity of both mitochondrial as well as nuclear genes. The ribosomes are similar to bacterial ribosomes, that is of 70S. All ribosomal proteins are synthesized by the nuclear genes and are transported to the mitochondrion from cytoplasm. The nuclear and mitochondrial genetic systems cooperate with each other to synthesize proteins and other main components of mitochondria. Mitochondrial growth is the integrated activity of both mitochondrial and nuclear genes. Therefore, though mitochondrion has its own genome, it is considered as a semi-autonomous organelle and not autonomous organelle, because mitochondrial protein synthesis or cell growth depends on nuclear genes along with mitochondrial genes. So, it is considered as a semi-autonomous body. Thank you very much.